working? Wonderful. So thank you all to SciLife Lab, to Science Magazine, and the Wallenberg Foundation for having us here. It's really been an incredible weekend and an incredible honor to be here. Oh, great, so this thing works. Okay, so we were asked to give you guys some background at the start of our talk about who we are and where we come from. So I was born and raised in Seattle, Washington, and after graduating high school, in Seattle, Washington is perhaps best known around the world for bringing Starbucks, Amazon, and Microsoft into kind of being. And then after high school, I flew cross country to the state of Massachusetts, where I've been ever since. I got my undergraduate degree at Williams College in Western Mass, and then after completing my studies there, moved to Boston, where I did my PhD at MIT and my medical degree at Harvard. And since graduating this past summer, I've been, as Mia mentioned, a resident at Brigham Women's Hospital. And so the work I'm going to tell you about today is the work I did during my PhD in the van der Heyden lab at MIT. Now, in the van der Heyden lab, we study the metabolism of proliferating cells. And it has been known since about a century ago, with the work beginning with Otto Warburg, that proliferating cells, such as cancer cells, metabolize nutrients and obtain nutrients differently than non-proliferating cells. And the reason for this is actually quite simple. Whereas a non-proliferating cell, its sole goal is to basically maintain homeostasis. It basically takes all the nutrients it takes in and turns them into energy, or ATP, to kind of maintain baseline. A proliferating cell, like a cancer cell on the other hand, has a very different set of priorities. Not only does it need to make energy, it also needs to make all the new components of a new cell. So proteins for structural reasons, nucleic acids to pass on information, and lipids for membranes. Now, most of the work done in cancer metabolism today has been done in a plastic dish in cell culture. And this has led to very powerful insights about how cells actually obtain these nutrients and make new versions of themselves. But it necessarily simplifies and ignores many complex interactions that happen in the body. So the prime example of this, so this is a PET CT scan using radio-labeled glucose. And what you can see here is these two sites are two metastatic disease sites from the same primary tumor. And you can see they actually take up glucose very differently. So there's very clearly something going on in this setting that you can't quite get in this setting. And so the goal when I started my PhD was basically to ask, can we observe changes in systemic metabolism that are revealed because we're using much more complex in vivo settings than you might otherwise see in a plastic dish. And then, of course, if we can find these changes, and hopefully we can find these changes, we can then ask what they mean and what they tell us about cancer biology. And so to begin this work, we actually started with pancreatic cancer. And the way we approached it is, And the way we approach this study is to perform what's called a prospective cohort study. And so we did this in collaboration with Brian Wolpin and his group at Dana-Farber Cancer Institute in Boston. And the way in which these studies work is you get, I'm pressing the wrong button again, ostensibly healthy individuals to give blood, and then they're followed clinically for a period of time, in our case, 25 years. And over the course of this 25 years, a given number of them develop pancreatic cancer. So we pulled these samples as well as two matched samples who did not develop cancer. And then at the end of the day, we decided to perform metabolic profiling and ask what, if anything, was different between the two groups. Now, before I get to showing you the data, I want to kind of highlight two important things. The first is that these prospective cohort studies are actually critical and a very nice setup, because one of the confounders you could run into if you actually profile individuals once they were already diagnosed with cancer is there's many confounding effects. You can imagine someone that's sick with cancer has different dietary intake, different metabolism at baseline, and so anything you find would be necessarily confounded by those variables. At the same time, though, a necessary caveat of doing a prospective study is that the individuals are not yet diagnosed with pancreatic cancer. So any findings, anything different you see at this end is therefore identified as a risk factor rather than a true true marker of disease. And this last caveat, even though it exists, is something you'll see we've challenged during the course of my PhD. And so these are the data. The metabolites we looked at are on the left-hand side, and they're not that important to read into detail. And going out to the right-hand side is a marker of the significance of the difference between the two groups. And three metabolites stood out to us. And these are a related group of amino acids called the branched-chain amino acids, isoleucine, leucine, and valine. And what was interesting, actually, is when we actually looked at these metabolites, the individuals that had the highest levels of these branched-chain amino acids were more than 2 times as likely to be diagnosed with pancreatic cancer as those individuals that had the lowest levels of branched-chain amino acids. So that's all good and well. And remember what I just told you is that because of this prospective cohort study, branched-chain amino acids will be traditionally identified as a risk factor for future disease diagnosis. Now, this didn't quite sit right with us as we looked at our data, and the reason is because of this. When we looked at our samples, the average time from blood draw to diagnosis was about 8.5 years. Now, there were some publications in the literature and kind of some rumblings within the pancreatic cancer community at that time that argued that pancreatic cancer was really present on the order of somewhere between 7 and 10 years prior to diagnosis. And so based on this kind of hypothesis, we decided to stratify our blood samples based on when they were drawn to when diagnosis occurred. And lo and behold, what we found is, if I press the right button, is that between 2 and 5 years prior to diagnosis, and even up to 10 years prior to diagnosis, there was a significant elevation in branched-chain amino acids in the individuals that got pancreatic cancer, or that were diagnosed with pancreatic cancer. So this to us really argued we probably were measuring true subclinical undiagnosed disease in these individuals, rather than predicting its future development. But the question is then, how do you prove that? And so to prove it, we turned to a couple different mouse models in the lab. Now, both these mouse models were driven by KRAS and P53 mutations, and I'm only going to show you one model here. But KRAS and P53 are two of the most common mutations in pancreatic cancer. And what we found when we looked at these mouse models is, strikingly, at times when disease was known to be present, you saw elevations in branch chains, but not before that time when disease was known not to be present, really arguing to us that in the human samples, we really were measuring true disease and not predicting its future development. Now, the next obvious question is, okay, great, we have these elevations, where are they actually coming from? And so in the Van der Heyden lab, we use a variety of metabolic labeling techniques, and so we actually performed these studies in our mice, and we were able to show that all of the elevation, the entire increase in levels of branch chains in the blood, was a result of a release of branched chain amino acids from the long-term protein pools in the body. Now, practically speaking, what are the long-term amino acid pools in the body? And practically speaking, they're actually really muscle. Muscle is primarily composed of protein and is kind of the huge body reserve that you activate to release amino acids into the blood. What we found when we actually looked at the skeletal muscle, in particular these mice, again, at the very early time points at which they had elevations in their branched chain amino acids, we saw a significant decrease in the size of these muscles, really arguing that we were getting a release of branched chain amino acids from intrinsic protein stores in these animals, and likely in the humans as well. Okay, great. So hopefully now I've convinced you that in the setting of a disease, in pancreatic cancer, we see systemic changes in metabolism. Now, one of the questions that kept cropping up in the course of our work was, how broadly applicable are these findings? Are they only unique to pancreatic cancer, or can they be extended to other forms of cancer as well? Now, actually addressing this across other forms of human cancer, or even many forms of mouse cancer, was kind of technically impossible. And so in order to kind of approach this in a more logical and more comparable way, we decided to focus on another KRAS and P53-driven model of disease, and that is, again, the wrong button. This is the non-small cell lung cancer model, as you see here. So again, driven by the exact same mutations, just a different location in the body. And one of the striking things to us, as soon as we actually did this study looking at the plasma, again, we confirmed the elevations in branch chains in the mice in the blood of the mice with pancreatic cancer. But then we looked at the mice with lung cancer and found that they had the exact opposite change. Now, this was striking to us. Again, same genes, but different location. And this was puzzling. Why would we actually see this discordance? And what we began to hypothesize is that in pancreatic cancer, not only were we getting a release of branch chains, but you might actually have the tumor not actually be using the branch chains. Whereas on the other side, lung cancer might be actually physically taking up these amino acids to use them. And so to actually prove this idea, again, we turn to metabolic labeling. And if you actually look at the tumors themselves, if you compare the control to the tumor, you again see a 2 to 3-fold increase in the amount of branched chain amino acids taken up by the lung tumors, which is consistent with this decrease in the blood levels. On the other hand, the mice with pancreatic cancer, the tumors in the control pancreas really show no difference in their amount of uptake. So this actually creates a bit of a conundrum. Remember I told you on one of my first slides that the idea of cancer is to proliferate and to make new versions of itself. Now, pancreatic cancer is still cancer, and it has to obtain the amino acids necessary to make the new proteins for a new cell. And if it's not actually getting them through free amino acids, well, then how is it actually getting them? And the short answer is through this process called macropinocytosis. And so what we found through, this is just a brief example, but basically pancreatic cancer cells take up bulk protein from outside the cell and then metabolize it internally to get the free amino acids. So they can actually ignore the free amino acids in the blood if they can obtain them this way. And this is just a brief comparison. On the left is the lung cancer, on the right is the pancreatic cancer, and you can see a much more dramatic uptake of this bulk protein from outside the cell. Okay, great. So now turning back to lung cancer. So we've shown you that lung cancer cells take up more branch chains. The next question is, what are they doing with them? And so on the left-hand side is a map of branch chain amino acid metabolism. And I promise I'll spare you the details. But the basic idea is we can use metabolic tracers and figure out where these branch chains are going in the cell and how the cell is using them. So the first obvious place we looked is the TCA cycle, which is how you generate ATP, energy, for a new cell. And so we actually fed these cells to labeled branch chains and then looked at citrate, which is the entry point into the TCA cycle. And what we found is that between the controlled lung tissue and the lung tumor, there was actually no difference in how much of the leucine made it all the way to the TCA cycle. So it really wasn't about energy generation for the lung tumors. So if it wasn't about energy generation, what was it about? And we decided to look upstream between leucine and the TCA cycle, and what we ended up focusing in on is this compound called alpha-ketoisocaproate, or KIC. Now, KIC is generated by removing a nitrogen from leucine, and it's the first step of branched-chain amino acid metabolism. And again, as you see here on the right, you see really a threefold increase in the levels of this KIC generation, which to us actually raised the question of whether or not there was something unique about this nitrogen that the cell was actually taking up the branched-chain amino acids to get. And so what would nitrogen actually get you in this context? And so nitrogen actually provides you with two key components for a new cell. The first is that it actually gives you nitrogen to make non-essential amino acids. So you can make other amino acids to make more protein for yourself. And the second is that it actually gives you nitrogen to make DNA, which is critical, obviously, for passing along your genetic information to your daughter cells. And so we can actually feed cells labeled nitrogen and ask, where does the nitrogen go? And as you can see here, we see significant increases in the lung tumors of how much nitrogen is being put on amino acids to make new amino acids. And we see marked increases in the production of new DNA. Again, really arguing that there's something important about the nitrogen in the lung cancer cells from branch chains. Now this raises an interesting and obvious question for us. If lung cancer cells are very particularly doing this, what happens if we actually block this step? So using CRISPR-Cas9 mediated technology, we actually deleted the two enzymes responsible for this process. And lo and behold, what we found is that you get a significant suppression, actually, the ability of these lung cancer cells to form tumors. On the other hand, and as you might expect from my earlier data, pancreatic cancer cells don't really care. They're not obtaining amino acids in this way, and so blocking the ability to make new amino acids doesn't affect them at all. And so this is actually fairly profound. Again, same genes, different locations, and we have very different effects. And the key point here is that typically the modern paradigm in oncology and targeted therapy at this point is that if you know the genes, so in this case we know the genes, you can know metabolic phenotypes and potential therapeutic vulnerabilities. Now this data would argue, however, that there's much more to this picture than you would expect, because clearly there's something going on beyond genetics and has something to do with the location in which these tumors actually arise. So the next question is, if the actual genetics of the tumor, the cancer genome itself, don't actually tell us what we can target, what happens if we look at another genetic approach, say expression profiling? And so this map on the left is kind of the first few steps of branched-chain amino acid metabolism. Again, the steps that are responsible for extracting nitrogen. And what you can see is you do actually see a modest difference between the control tissues and the tumor tissues for the lung and for the pancreas that might actually help you predict the signature. The only problem here is, though, is that you're looking at about 3 to 5 genes between the hundreds and hundreds, if not thousands of genes that actually differ between pancreatic and lung cancer. And so to actually pick out the signature, we think is actually fairly far-fetched. The expression data does raise an interesting question, though, however. And the question there that comes out is, are these profiles, as I just showed you, actively selected for in the tumor, or are they merely a byproduct of the location in which you came from? And so we can actually ask that question a little more formally. We can actually make the pancreatic cancer cells, the ones that don't care about free branch chains, care about free branch chains by overexpressing the transporter that allows uptake of branch chain amino acids. And so we can actually make these cells take up 2 to 3 times more branch chains. And then when we actually put these cells back into mice, we can see it doesn't matter. And so what that to us argues is that it really isn't a selection against this process in pancreatic cancer cells so much as just kind of a natural continuation of where they come from and what they choose to do based on that reality. And so then taking a step back, where does that really leave us? So precision therapies in general are at this point based on targeting known mutations. But hopefully I've convinced you by now that it's really not so simple, that there's really a significant contribution based on where you come from and who you are and your lineage that significantly shapes your vulnerabilities, especially when it comes to metabolism. And this has actually already been borne out to some degree in that our most successful metabolic therapies in cancer are things like folate antagonists, which are actually chosen based on tumor type rather than your genetic profile. So the question then is, how does this model exist in practice? How do we put it all together? And the way we like to think about it is that basically every cell in the body has a generic set of all metabolic pathways that are available to it. And then these pathways are then plunked down in different locations in the body in a normal cell. And the normal conditions in that cell basically constrain the metabolic pathways that are available in that location. Genes then don't actually create anything new. They're superimposed on top of this existing constrained network and can help shape that network and kind of alter the way in which things move in the cell. But they can't recreate it de novo. And indeed, this actually makes sense. If you're a cancer cell, why would you create something from scratch when you can just hijack what's already there? And so the hope and the goal of this project really was then to get us to think about, if we're going to actually do precision medicine in terms of metabolism, we really need to use effective models that can reveal these vulnerabilities and these biases so that we can actively target our approaches and our therapies moving forward. And so with that, I have a lot of thanks to people to give, primarily my advisor, Matt Vanderheiden, and then as well as Megan Torrance, who's an undergraduate in the lab who worked with me for 3 years and basically had a hand in almost all the data I showed you. Also my collaborators at MIT and at Columbia for help with some of the computational work. And then the human cohort work was only possible really because of the efforts at Dana-Farber and the Broad Institute as well. And with that, I guess I'm taking questions.